Hello and welcome, everyone. I'm Peter Stern, one of the editors of Science Magazine, and this is our first in-person conference that we have organized together with the Chen Institute. We've had a number of conferences before in the past, but because of the pandemic, they all had to be online only. However, they were extremely successful and we had really thousands of participants. So we thought, now the pandemic is over, the neuroscience meeting is in town, let's go and have an in-person meeting. So welcome to everyone here in the room. I was told that a lot of people are still stuck in traffic, so I assume that in the next couple of minutes there will be more joining us here in person. And also welcome to everyone online. I was told that we have about 1,000 people who are participating from all around the world. So over the last years, our collaboration with the Chen Institute has been extremely successful. And we had a number of conferences on topics like human cognition or brain-machine interface or neuromodulation tools. And so we were always just bang on. This was topics that were widely discussed in the community. So this year, our topic is AI and mental health. And I tell you, we came up with this idea a little before the latest of the waves of AI being in the public domain and being discussed everywhere. So we are proud that we just smelled right in time that this is something that's going to be big. So without further listening to me, I think we will probably start with our speakers. The first speaker this morning is Mandy Wang. She's based at the Center for Statistics and Machine Learning at Princeton University. And the title of her talk is Diffusion Model for Guided Generation with Bioapplications. Mandy, please.
Okay, thank you for the help. And Peter, thank you very much for the nice introduction. So, it's always very hard to get back to in-person teaching and in-person conferences since the pandemics. So, I have collaborators who we wrote many papers already, but we have never met. I even had a postdoc who we have worked for 3 years. And he's now a faculty member in Singapore, and we have never met. And now that he is in Singapore, I feel like maybe we just never get to meet. So this is really the norm now, with the hybrid of online conference and in-person conference. But I'm happy to be here today in person. And also, I was in the morning I was taking a cab, and I definitely know how bad the traffic is. Okay, so I'm going to talk about diffusion models, which is a particular powerful type of generative AI model. I'm going to talk about what's going on there and hopefully to showcase some potential applications in biosciences. Okay, so when we think about generative AI, I think the two most common applications that we actually use every day that has become part of our life is ChatGPT and actually DALL-E. Okay, ChatGPT is an autoregressive model for text generation. But other than text generation, if we think about generative AI when we try to generate continuous data or continuous objects, for example, images, or even continuous control policies, or even protein structures. Okay, so if we think about generating something that is continuous in nature, so actually the fundamental model for the latter three applications are diffusion models. Okay, so autoregressive for discrete text and then diffusion models for continuous generation. So those two are basically I would say the two pillars of generative AI today. Okay, so diffusion models, I mean, the two major players, one is the Stable Diffusion and the other one is OpenAI. I mean, DALL-E 2 is actually outdated. Today we're using DALL-E 3. So if you play with it, if anyone hasn't played with it, I strongly recommend you that just enter on your browser, enter OpenAI and click DALL-E 3. Okay, so you can enter a prompt, and the prompt and the Stable Diffusion or this DALL-E 3 is going to just give you a number of images, try to match, try to align with your prompt. So this is really cool. And at the same time, we also see a lot of news that artists are really angry about this because of this patent, I would say privacy leakage and also IP issues. And some artists worry that they're going to lose their job, and other people feel like, okay, this is really just a recombination of things that have already been there. There's no novelty. Anyway, so there's a lot of debate there, but I want to say that fundamentally, the underlying model of this is diffusion models. And they are extremely important for not only image generation, but they have showcased very immense potential in other applications. Okay, so let me discuss a little bit about what's going on when we talk about generative AI. So where does this come from? So I guess the initial sparkle of generative AI maybe come from autoencoders. Okay, so autoencoder is a neural network architecture with an encoder and a decoder. So the encoder is considered as a neural network mapping a high-dimensional vector to a low-dimensional vector. So it's basically compressing data into something more concise. And then the decoder tries to start from the low-dimensional vector and reconstruct the raw data, maybe an image or other objects. Okay, so autoencoders or variational autoencoders, VAE for short, they used to be deep neural networks for compression and reconstruction. Okay, but then ever since this encoder-decoder architecture came into being, then people realized that, okay, if we only look at the decoder, if I already have a decoder, right, now we can just draw a random Gaussian noise as input and try to see what we can decode from the random noise, right? And that's the first time people realized that, okay, this architecture can do something more than compression. It can actually do generation. And then this become really prominent with GANs, generative adversarial networks, which in mathematical language, essentially they have a primal dual, I would say a duality architecture with one generator and one discriminator. So the initial goal was to do classification. So initially the focus was to get a good discriminator. And then people realized that this generator from noise to data, but that generator actually does something very unexpected. It's actually more interesting than the discriminator. Okay, so now the focus has shifted to that, okay, we really want to think about generative AI, meaning that we want to find a neural network architecture or any generation process so we can start from noise and magically decode from noise into data or decode from noise into novel designs. And then the state of art, which is basically the foundation model for all continuous generation. So the state of art is diffusion models. Okay, so let me just mention a little bit about the idea of diffusion model. So the idea is actually very conceptually simple. Okay, so the basic idea is, you know that we want to start from noise and then transform noise into data. That's definitely a very hard problem. Okay, but if we reverse it, if I start from data and then make it into noise, everyone knows how to do that. Right? I can tell you a million ways to jeopardize my data and make them into noises. Okay, so let's consider a very simple way. Let's just add data, add Gaussian noise continuously to data until my image, maybe my image data become completely blurry into white noise. Okay, so why is this process interesting at all? Okay, this is a stochastic process that we want to reverse. Okay, So this forward process, we just add the noise. And we can pick any differential equation we like and add Gaussian noise to it. So here we're just picking a very simple one. So that negative 1/2 is only for normalization, so that we make sure that all the variances stay constant or bounded. Okay. Okay, so now we have a process of adding noise, right? So that's very easy. We can simulate this process very easily. I just sample images and then add noise. Okay. So now the real question is, can we reverse it? Okay. So here we're actually using a notion from a stochastic process about trying to figure out the time reversal of a stochastic differential equation. Okay. So we can imagine that, so what we really want is this backward process. So we start from noise. Okay, so we have this initial distribution, which is a Gaussian process, and we want to define a stochastic diffusion process. So from noise, we eventually reconstruct data. Okay, so then the idea is, okay, I want the first time step of my backward denoising process to match the second to last time step in the forward process. By matching, we mean that we want their marginal distribution to match. Okay, and then we move on to the second time step, which should match to the previous second to last time step. So we keep doing that. So overall, the problem is that we want to find the transition function for the backward diffusion process such that all the marginal distributions of this backward process matches nicely with the reverse order marginal distribution from the forward process. Okay, and this is actually a very neat mathematics. Okay, so what's going on is that one can actually derive a closed form transition function for the backward process, which is what we call the score function, which is actually the gradient of the log likelihood of the marginal distribution with respect to data. Okay, but still, I mean, we know that the score function should be that, but that function is not something we have. Okay, so training diffusion models is about training a neural network to match that score function. Okay, so there are so we work on machine learning theory and algorithms, so we actually care about theoretically, can diffusion models actually learn the underlying data distribution? And secondly, we want to see, okay, can we use diffusion model in a way to do better generative AI? Okay, so the key here is to learn the score function, which is the gradient of log marginal likelihood. Okay, so by learning it, we mean that, okay, we're going to fit a neural network. Okay, let's consider, I would say, a simplified version of a score function. Here, we still have an encoder and decoder, and we have nonlinear transformation in the middle. But we can imagine the encoder and decoder, we can imagine that them as, for example, the encoder, we can imagine it as a very tall matrix, okay, which would map high-dimensional image to low-dimensional embeddings. Okay, and in typical architecture of score network, usually there's a shortcut. We can imagine this is like a simplified version of the ResNet link. Okay, so based on this architecture, we actually can show in mathematical theory that this diffusion model, which is learning a sampling process, so this diffusion model is actually implicitly a probability density estimator. So it actually learns the underlying data distribution. And further than that, it is not only a density estimator, it is a statistically optimal density estimator, meaning that with this statistical function class, diffusion model is already learning some sort of a sufficient statistics. That's the best approximation one can get based on finite time data. So that's good. Okay, so this generative AI model is actually a probability density estimator. Okay. So secondly, so diffusion models actually can do much more than just learning a distribution. For example, when we actually try to generate images, we're going to generate images conditioned on the user prompt. So my prompt can be say that, oh, I want you to generate a dog or a cat, or I want to generate the the headquarters of AAS. Okay, so that will be the prompt. Okay, so what's going on in this backward, forward diffusion processes? This prompt is what is being conditioned on in a probability space. Okay, so here the problem becomes that, okay, I'm trying to generate or I'm trying to learn a probability density function, and that is not just a marginal density. We're trying to learn a conditional density function. Okay, but the only difference here is that when we try to learn that score function, now we're conditioning on y. Y, consider it as a prompt, okay, or just anything we want to condition on. And this turns out to be universally very, very neat to implement and very powerful, because we're only modifying this score function a little bit. Okay, so that modification, usually we call it guidance. It means that one can have some external information to guide the generation process. So that little y there plays the role of guidance. Okay, so that can actually help us to customize a big pre-trained diffusion models to generate something we actually want. Okay. So on top of that, because we're talking about generative AI and also especially applications in like biotech, okay, usually we want to consider design problems or even optimization problems. Okay, so in the language of optimization, let's think about, say that I want to design, I don't know, a sequence of amino acids. And maybe I want to design a protein encoding sequence so that I have some nice binding affinity. Okay, so if I put my hat as an optimizer, I would say that, okay, I want to maximize binding affinity, which is a function of the amino acid sequence. Okay, so I have a finite function f of x that I want to maximize. Okay, so if we think about this as an optimization problem, we are going to be screwed, basically. Okay, because the space of potential solutions is too big, and we really don't have a good way to handle the underlying structure. Okay, but now let's think about the problem as a generative AI problem. Okay, so what if my problem is that I want to draw samples from a distribution of X conditioned on that my finite value is high. Okay, and now we can utilize the architecture I just described. We can try to, I would say, start from noise and transform noise into real data or even novel data. Okay, and then we can use diffusion models. Okay, so here, my goal here is that we want to use diffusion models to solve optimization problems that cannot be solved using traditional tools. Okay, so this is one example very recently. So for example, in order to just generate better images that better align with user preferences, one can try to have a reward function to measure the level of alignment between prompt and images. Okay, and then that can be an additional optimization objective. And then you can see that one can generate images that better align or more realistically depict what the user wants. And further, we can choose the reward function to be maybe an artistic style. Then it means that during this generation or denoising process, we can just add a little bit of tweak to make sure that we're generating something that's of a particular style. Okay. Okay. So people actually also do this for protein design. So this can be done in both the sequence space and and the structural space. Okay. So here the problem is one may want to maximize some biochemical property of the protein. But if we translate it into the language of conditional generation, we're going to say that condition on that the property is nice. Can you generate new designs of X, the protein encoding sequence or the protein structures? Okay. Okay. So like coming back to this I would say to this roadmap, right? We want to make a connection between the two, but a huge problem is that when we solve an optimization problem, okay, we always care about the curse of dimensionality. Okay, so comparing the problem of optimization against learning a full joint probability distribution. Okay, so actually, if we are just comparing those two, approximating a distribution is actually a much harder problem, even from the perspective of information theory. Approximating distribution is just harder because we are trying to learn something more complex. But for optimization, we only need one single solution. Okay, so in some way, optimization is simpler, right? But then why do we want to go into this additional trouble of training a diffusion models? And what can we gain out of it? So that's the question we want to answer. Okay, so the short answer is in any real data or in any real design problems, we have a lot of like geometric structures. Okay, so this is very intuitive. Say that, oh, I have a data set which are just photos of a teapot taken at different angles, right? We just walk around the teapot and take pictures, right? So the degree of freedom in this data set is only one because we are just moving and there's only one angle there, okay? And even for like human facial expression, okay, for a single person, we need around only 3 or 4 coordinates to represent his or her facial expression. Okay, so what does it say? It says that, okay, the number of pixels, which are the raw dimension of raw data, they can be huge or very large, but really the data lives in some very simple low-dimensional subspace or manifold. Okay. Okay. And this is a very, very nice paper this year. So this paper is trying to examine all the public benchmark image datasets and try to approximately estimate the intrinsic dimension of the dataset. So they work by using a nearest neighbor approach to construct a big adjacency matrix, and then they do some graph decomposition to estimate an approximate dimension. Okay. So basically for the biggest dataset there, ImageNet. Okay. So the dimension is around 40, meaning that for all the all the images in ImageNet can be approximately embedded into a subspace of dimension only 40. Okay. Again, this is that data have a lot of structures, and data are low dimensional. Okay. Okay. So this is really what's going on here. So in our own research, we try to study diffusion models under the assumption that my data X, considered as an image, can be embedded into a low-dimensional vector. Let's consider it as a small vector Z. We think of Z has dimension 20 or 40, but X can be an image of dimension like 1,000,000. Okay. So what's really going on is we can imagine like in that picture, so my data can be 3-dimensional, even higher dimensional, but they live in a rotated, distorted manifold. Okay. So if we can like flatten out that manifold, then my data becomes really easy to work with. Okay. So that's what's going on here. So what's interesting is in diffusion models, as I explained, we have noising process and denoising process. What's going on is that if my data truly live in a lower dimensional space, then the diffusion process naturally decomposes into a process in the low dimensional space and another process in the orthogonal component. Okay, and turns out that orthogonal component does not matter. The concentration of the process really happens in the lower dimensional space. So we were able to establish the first theoretical proof that diffusion models naturally adapts to low dimensionality of data. And the sample complexity of learning from data scales with the smaller dimension, 40, rather than the bigger dimension, like 1,000,000. Okay, so this is great. Generative AI models, this is just an example, but generative AI models, essentially they're learning the underlying latent low dimensionality. That's why they are powerful. Okay, so we further extend diffusion models to try to do optimization, because I told you we want to do optimization. Now, when we do optimization, okay, so we are going to, usually we work with a large unlabeled data set. Imagine the set of, I don't know, like all images or the set of all RNAs, right? But we don't have or do not have label. Okay, the diffusion models can be pre-trained on those data sets. Okay, and then we have some fitness function, maybe binding affinity, or maybe some user preference, right? But for those fitness function, we usually only have a smaller labeled data set. Okay, so we're going to learn from the combination of labeled and unlabeled data. Okay, so we're going to try to learn the reward function, which is the objective of optimization, by reward learning, by fitting from the labeled data. Okay, and then with our reward model, we can use it to annotate data and generate pseudo labels. Okay, then the pseudo labels can be further utilized to guide a pre-trained diffusion model so that it can generate something that would maximize the reward that we hope to improve. Okay, and finally, with this, I would say, fine-tuned or guided diffusion process, we can do generation of novel designs. Okay. Okay. So again, theoretically, what's really interesting, I don't want to bother you with all the mathematical equations, but theoretically what's going on is this particular diffusion model is actually better than any existing optimization solver. Okay. Further, it's solving the problem implicitly in the low-dimensional space. And if I compare it with an optimization solver that knows where the low-dimensional space is, okay, even if I tell the optimization solver to work in the correct low-dimensional space, this diffusion model is still a better optimizer. Okay, so that's really nice to have. Okay, so we did a lot more experiments. For example, if we try to generate images, okay, so this is about generating images and try to improve, I would say, a reward function that measures this contrast and the colorfulness. Okay, so one important thing to notice that as we try to guide the generation process, right? So we're essentially extrapolating from training data. But if we do extrapolation too much, then the quality would actually degrade due to that the model becomes out of distribution, which is a very important concept in all deep learning. Okay, whenever we extrapolate too much, we're going to get out of distribution. So one needs to be really careful and when we do de novo designs. Okay. So we also applied this approach to continuous control. So here the idea is super simple. So in control, usually one has to solve Bellman equation and call a very complicated reinforcement learning algorithm. But here the idea is simple. So say that we have a bunch of labeled state trajectories. Imagine that they are like trajectories of a robotic arm. Okay. And then we're going to simply train a diffusion to learn from those data. And then I say that, okay, my diffusion model, now you are my magic mirror. I want to have a trajectory of very high reward. Can you imagine such a trajectory? Okay, so what happens is diffusion model can imagine a state trajectory with actually higher reward. And then one can do reverse engineering and figure out to get such a trajectory, what should be the sequence of controls or actions. Okay, so we actually benchmark our algorithm with the best reinforcement learning algorithms. Okay, so it turns out that as long as we're extrapolating at the right amount, these diffusion models, which completely ignores the control nature of the problem. Okay, for a purely generative diffusion model, it actually performs comparable, even better than the best reinforcement learning algorithm. Okay, so that's about diffusion models. So I want to spend maybe like 5-10 minutes talking about a close analogy of diffusion models, but in science and nature. Okay, so remember that in diffusion model, we are adding noise and then denoise and then guide or tweak the process. Okay, so what's really similar to generative AI in biology is actually evolution. Okay, so one, I would say, extremely important breakthrough in the last century in biotech or biology is directed evolution, which marks like the first time, it was in 1950s, but the Nobel Prize was awarded later. So it marks the first time when humans can artificially evolve protein variants. So they can generate new protein variants in the wet lab by inducing mutations and then doing like artificial selection. Okay, so the idea is super simple. So say that we have a plate of variants, right? Consider like a plate of cells. Okay, and then say that I have some chemical compound that I can add to them, and then they are going to start mutating and recombinating. Okay, and then like after 2 days, we're going to screen my variants. So some of the mutations, most of the mutations are going to be garbage. We're going to throw them away. But some of the mutations are going to be nice, and we're going to keep them. Right? So this is evolution, right? And if scientists try to control the process in a more artificial way, this is called directed evolution. Okay, so there are tons of methods in this domain, some purely wet lab, some more in silico. Okay, but this is really how scientists are evolving proteins. This is how therapeutic companies are designing drugs. Okay. Okay, so in directed evolution, so there are two typical operations for adding noise. Okay, this really is similar to the forward process of diffusion models. Okay, so one is just mutation, right? We know mutation, like some random sites of a sequence randomly get flipped to another token, right? So this is pretty much like adding white noise, although we are talking about discrete sequence. Okay, but another very powerful operator in direct evolution is recombination. Okay, it means that we can have like two parents and then they can like cross over and generate children. Right. In certain scenarios, we can actually artificially induce DNA shuffling, meaning that we have a bunch of sequences and then we're going to just disassemble them and they are going to reassemble randomly. Okay, so those two, I would say we consider them as an analogy of adding noise in the forward process. Okay, but this is what we already have in nature. Okay. And in the recent, I would say 5 to 10 years, I mean, using machine learning and AI to accelerate direct evolution, this has become this really big thing in protein design, and even more than protein design. Okay, so this was actually I would say a review paper by the Nobel laureate Francis Arnold in 2019. So they surveyed how biologists and scientists are trying different, I would say, machine learning heuristic in their actual wet lab design. Okay, so and today with generative AI and with all the tools we have, we want to understand this more principally, and we want to have a provably efficient approach. Okay, so how are we going to add machine learning to direct evolution? Okay, so the process works like we have an initial population, and then the population will diversify by mutation and recombination, right? And before we actually run a wet lab screen, we're going to use a machine learning algorithm to do important sampling so that we have a virtual screening aided by, for example, a deep neural network. Okay, so that's really the only way we're going to use machine learning here. So here you can imagine that this overall process is a big diffusion process. Okay, but however, most of the steps happen in the nature or they happen in the real world. It's not like they are not happening by computers. But we can only use our AI model to change one single step in this physical or this biological diffusion process. And can we utilize our minimal machine learning intervenes to accelerate the process? Okay. So you can imagine that this machine learning selection step is going to be very nice because it can help us to rule out variants that are obviously not good, right? So when we run our actual experiment, we're going to use our throughput to evaluate candidates that have more potential. Okay, So again, so if we think about this as an Bayesian machine learning problem or as an optimization problem, the problem is really hard. Okay, so in optimization, what happens is we never have a good estimate of the fitness function. For example, the binding affinity function. So one would have to have some neural network to represent a posterior distribution over the possible choices of the fitness function. Okay, and then within that posterior distribution, for example, one can come up with confidence regions and then try to generate samples with higher uncertainty to learn better the function while optimizing. Okay. But however, this is an approach that works very nice in a computer. It doesn't work in direct evolution because we simply cannot easily compute the maximization of a function over my space of biological variance. Okay, so basically we want to utilize this Bayesian machine learning idea, but use it to tweak the biological diffusion process rather than following, I would say, the formulas exactly. Okay, so this is what's going on here. So basically, we can imagine the top row is what's happening in a real lab, right? So a population has its mutation and the recombination, and one can run screens to do selection, right? But at the same time, we have a machine learning model here. So this machine learning model, we consider it as a Bayesian model, which keeps getting updated whenever we have new data. Okay, so we can think about this a posterior model or a big pre-trained model that is being fine-tuned with new data. Okay, and then my machine learning model is going to I would say guiding this forward diffusion process by saying that, hey, this sample is not good, give me another one. Okay, so we're actually tweaking the distribution of variance by using the machine learning model. Okay, in some way, this is actually very similar to backward generation in diffusion model. But now we're talking about a real wet lab process. Okay, so this is a very simple simulation. You can see that initially we start from a solution which is wrong. Okay, and then the population will diversify because of mutation and recombination, right? And while during this process, our model is also learning better about this protein and its function, right? And then this guidance will play a role, and you can imagine the population will initially diversify and then move towards the optimal solution and concentrate. So this entire process, we can think about it as we're first adding noise and later we're doing backward denoising when we have a better guidance. Okay, so this turns out to work very well. So we try this out on some, I would say, benchmark protein systems like G1. It can get a near optimal, very good solution using only 200 variants, by measuring only 200 variants in total. Okay, so I have only 2 more slides to go, so I'll be on time. So this approach applies to more than protein engineering. So I got into a very interesting problem about CRISPR gene editing, which we're going to use this generative AI model to design. So like gene editing, usually we want gene editing to be precise. We do not want offsite errors, right? But there is a particular application of gene editing in which we actually want more errors, the more the better. So this is an application where gene editing is not going to edit the cell's own gene. It's going to edit an inserted snapshot, an inserted sequence, which does not encode anything. Okay, we can imagine this inserted DNA sequence is a barcode. Okay, which is common. I mean, people do this DNA insertion as barcode for single cell analysis all the time. Okay, so how can gene editing help? Say that I have a gene editing system which has a lot of errors. Okay, so from this initial barcode, this initial DNA sequence, gene editing is going to work, right? And then as the cell branches into 2 children, editing will continue in the 2 children. And the editing will continue to generate errors. So that means that actually we have a barcode that is dynamically evolving. And for two children, the evolving process become independent. Okay, so this is super cool because we only need to edit the parent cell, and that editing errors will keep evolving as the cell branches. And we're going to have a unique barcode for each descendant. Okay, and then by looking at this, I would say, genetic similarity between those barcodes, one can precisely recover the lineages. So we know who's the parent, who's the child. One can know about the entire process. So this actually means that we're having a way to add a time step to single cells in their, I would say, in their branching process. Okay. Anyway, so that's a story, but the design problem is we want to design a guide RNA that can induce a lot of errors. So mathematically speaking, so we measure the capacity of barcoding using the entropy of editing errors. We want to maximize the entropy. Okay. Okay, so we utilize a pre-trained guide RNA model as the basis, then apply this, I would say, AI-guided direct evolution. So in the end of, and then we work with a wet lab, so they actually try out the guide RNA designs and try out and measure the barcoding entropy. So in the end of the day, so we actually found a collection of barcode that improves the efficiency by more than 30%. And what it can actually does is that it allows, so this barcode will keep evolving, right? So the evolving barcode allows us to start from a single barcode and single root cell, and it can track more than 1,000 descendants, meaning that it can generate more than 1,000 unique barcodes. So we can actually track a big cell lineage tree by using this barcode. Okay. So just one more minute. So another similar project which actually uses language model is we try to understand the untranslated region of mRNA. So in mRNA, so we have this coding region CDS that will be translated into a protein, but the untranslated regions UTR, they are regulating the translation process. Okay, so actually in, like, for example, COVID vaccines, the efficiency of COVID vaccines are determined by the two UTRs, because COVID vaccines take effect because they can be translated into proteins. If they don't get translated, they are just mRNA in our body and they do not do anything. Okay, so we want to actually design the untranslated region of mRNA, but it turns out there's actually little tool one can use, and there I couldn't find pre-trained models. Okay, so we trained our own language model for mRNA. Okay, so we curated data sets, basically mRNA sequences, especially with a focus on the untranslated region from multiple species, including humans, mouse, fish, and other. Hopefully, because if we want to train a big foundation model, we need, basically the more data, the better, because we want to learn some common pattern about those mRNA sequences. Right? So we have a language model, and then based on the pre-trained language model, we further do this guided direction and generation. Okay, so eventually, so we were able to come up with a very small library of good novel designs, just sequences of mRNAs. Yeah, so we work with a collaborator who was kind enough to actually test our designs. So our best design actually outperforms the mRNA used in the COVID vaccine by 30%. So, which seems to be good. Which seems to be telling me that, so this language model approach together with generative AI actually can learn and generate something useful. Okay, so that's my last slide. Thank you very much. Okay, right on time.
On the bottom, just hold it down for a moment.
That's it? Okay. So thank you very much. That was quite a wonderful opening of our session today. Let's take a seat for a moment. And there's another microphone for you. And I would suggest we first ask people in the audience to come up with questions. And there will also be questions from our online audience. But let's start with the people in the room first. So please, the floor is open.
My question is, in this application for decoding and novel design of mRNA, what is the objective? I mean, when you say it outperforms, or I didn't get a sense of what it was that the sequences were being designed for.
Translation efficiency. For example, one metric may be just ribosome loading. So there are multiple metrics that correlate with translation efficiency. So here we're using one specific metric.
And that's like related to the pre-trained foundation model by virtue of fine-tuning, or how is the
we have a pre-trained models, and we also have some label data, for example, with ribosome loading measurements. Right? So we fine-tune the pre-trained model on those labels, and then we have a better pre-trained models. Right? Then we use the model to generate new designs.
I see. And so just to make sure I understand, the fine-tuning process is the conditioning method that you mentioned earlier, or what is the nature of the fine-tuning?
Okay. For this project, this is not a conditional diffusion model. This is more language model plus directed evolution. So this fine-tuning, so this is actually a language model. This is not a diffusion model, because we are talking about, I would say, mRNA sequences, which are discrete in nature. So then this language model, because it's common to use a language model, I would say, as an initial feature map. Right. So once we have an initial feature map, we can definitely fine-tune it on functions that we want to predict. And then this initial feature map is going to be used to construct a prediction model that will guide the evolution process.
Okay, thank you.
Thank you.
So there are microphones here. So if someone has a question, please come forward and go to the microphone in the room. Yang?
I was thinking maybe I will take, so some questions from online and pass to you, Mengdi. So there's a couple of questions. And the first one is about noise. So you talk about the forward and the reverse process introduce the noise. And the audience was asking, and how you control the noise in the process.
So the forward process is mathematically called an OU process, meaning that it's a process with a natural contraction. And then the noise is just Gaussian noise. So we can just have one single parameter tuning its variance. But the process is designed in a way to be contractive so that the limit distribution will be finite rather than go unbounded. So that's sort of how the forward process is built to have that property.
And the second question is relates reinforcement learning. So you talk about reward process and compare the diffusion model with reinforced learning. The question from audience, so they're asking, so can diffusion model be integrated with reinforced learning and to enhance the outcome?
Yeah, that's a very good question. So this is an active area of research. So we know that diffusion models can already work better in some particular problems of reinforcement learning. But on more complex problems, people are actually trying to utilize diffusion model, for example, as a world model to help, for example, policy gradient algorithms. So there's a lot of active research, very promising area.
Okay. I think the last one is related to your second part. So they are asking, can you share more insight about how machine learning and improve the accuracy of generating mutations and guide the predictions of direct evolution?
More insights.
Yeah.
Okay, so I think one necessary component is to start with a nice pre-trained model, actually. So pre-training is very important because we have to start with a good feature representation. And only when we have a good feature representation, maybe to learn a new function, we might always have to collect new data, right? But if we have a good representation to begin with, that means that we have already narrowed down the space of possible functions. Then we'll be utilizing data to run search within a smaller function class. So a lot of the, I would say, the recent advancement come from that people are pulling data sets together to have a pre-trained model. And then that pre-trained model turns out to be able to help many specialized individual tasks. Thank you.
One question I had, a number of the examples that you showed were more or less from the same areas of biology. But of course, biology is a gigantic field. So now I want you to stick your neck out a bit. Where do you expect, where do we see progress in the next years? Which other fields of biology will probably benefit most?
Mm-hmm. Well, I guess two major breakthroughs. One is it's really about measuring neural signals and neural imaging. So I think that is really taking off, and there are, tons of data out there already, but still there hasn't been too much, I would say, AI for neuroscience research going on. So another promising area is spatial omics, because that really allows people to look at, I would say, cell state transition with a much better measurement tool. So I think those two technologies are going to generate a lot of data, and hopefully we can learn from those data. Yeah.
So, are there any more questions here in the audience? So, we are exactly on time. Thanks again, Mandy. I think that was a very good opening.
My pleasure. Thank you.